I will have a presentation from Gabriela Castellano, professor from Unicamp. You can start.
Okay. So, good afternoon, everybody. My name is Gabriela Castellano. I am from Unicamp, which is University of Campinas. I will talk about complex networks to help understand brain organization or reorganization in motor rehabilitation and brain-computer interfaces. So, a disclaimer here is that my work is very applied, it's very different from what you have seen in the previous talk. I won't show many occasions, I will talk more about direct applications. So I am from Unicamp, so this is an aerial picture of University of Campinas, for the people that don't know it. The logo of Unicamp actually is based on this spatial distribution of the main campus. I was hired to teach the medical physics course that we have at University of Campinas that started in 2003. I actually started at Unicamp in 2005, and together with Professor Covolan, who is now retired, we founded the neurophysics group. And so when I first started, I used to spend most of the time here at the Physics Institute, but also one day of the week I would be here at the hospital, which is all this area here is the health sciences area of University of Campinas. And I would stay one day of the week here working at the MRI scanner. Then I left that for my students. And after the pandemics, we sort of didn't go back much there. But anyway, so I've been working in this area that is basically the study of the brain since I entered Unicamp. And I participated in very large projects that involved lots of researchers. The first project was called Synapse, from Cooperação Interinstitucional de Apoio à Pesquisa sobre o Cérebro, that involved the main universities of São Paulo. So we had USP, Unicamp, Unesp, and other institutions to study the brain with different techniques to study the brain, and focus mainly on stroke and epilepsy. Before this project ended, I started working in another project called Destiny, which was sort of development of IT for neurology that was based on developing assistive technologies for stroke patients. Then when this ended, the people of Unicamp from this project started the project BRAIN, which is a very large project from FAPESP, which means Brazilian Institute of Neuroscience and Neurotechnology. Now we are in the second phase of BRAIN, second and last phase of BRAIN. In the meantime, we had this other project called Extreme, which is experiences of technologies for rehabilitation in medicine. And now we are about to start this other project called DREAMS, which is development of rehabilitation and assistive devices for neuroscience and neurotechnologies. So as you can see, I have always worked with people very good at making acronyms for projects. So the BRAIN project involves a lot of areas. From all these areas, I have worked in these years. But today, I'm going to talk especially about these, or actually, I'm going to talk more about evaluation of assistive and rehabilitation technologies, mainly in terms of the brain changes that we can find, and specifically using complex networks. So for that, I'm going to talk first about the brain. So the basic structure of the brain, you can divide it in like some big chunks that are the lobes. You have the frontal lobe here, the parietal lobe, the temporal lobes that are in the sides, the occipital lobe. And we all know more or less what these lobes do. So this is the last lobe to evolve. So the more complex the organism is, the larger the frontal lobe is. And so it's responsible for more complex functions. The occipital lobe, for example, is mainly responsible for processing vision. And so on. I'm not going to talk about all the lobes now. This is an MRI picture of the brain. You can look at the brain also, instead of separating it in lobes, you can separate it it in two main tissues that are the white matter, which is actually the white part here, and the gray matter, that is all these parts here. And so the white matter is made basically of the axons of the neurons, and the gray matter is made mainly of the bodies of the neurons, or the soma of the neurons. We have about 86 billion neurons. Before, people believed that we had 100 billion neurons, until a Brazilian group actually went there and actually counted the neurons, and they found 86 billion neurons. Most of the neurons are actually in the cerebellum here, and only 16 billion are actually in the brain. And we also have in the brain 85 billion glial cells, which were believed to be cells of support of the neurons, but now they are known that they also have some information role. So this is a picture of a synapse, just to know. So the neurons to communicate, they will communicate through a chemical gap here. And for this, we need energy. And the energy to the brain is about 2% of the body mass, but it uses about 20% of the body energy, right? And we have basically two main ways of getting energy to the brain, but both ways are happen through the blood. So one way is the glycolysis, which is not very effective, is faster, but it's not very effective. We can get 2 ATP. And this is the anaerobic metabolism. We only need glucose for this. And the other aerobic metabolism, which is more efficient because we get another 34 ATP, needs oxygen and glucose. And so for the brain to function, we actually need a lot of blood and a lot of very dense capillary network structure. So I'm talking about this because to measure what the brain does, we have several techniques. I'm only going to talk about 2 techniques here. One is magnetic resonance imaging. So magnetic resonance imaging is basically based on the fact that some nuclei have spin, and if they have spin, because they are charged, they have magnetic moments. If you have nuclei with spins in a place without a magnetic field, they will be pointing pretty much everywhere. The magnetic moments or the spins, they are parallel, will be pointing everywhere. If we think about the human body, we have 10% of hydrogen in the human body, and the hydrogen has a nuclei with a spin, which is the proton. And therefore, if you get hydrogen in a place with a magnetic field, you can think that you'll have some of the nuclei pointing towards the magnetic field and some pointing against the magnetic field. But we will always have, at environmental temperature, we'll have more pointing towards the field, and then we get this magnetization. And they actually will make this rotation, this precession around the field, because they have the spin. And this happens with a particular frequency that we call the Larmor frequency. And if you have something that has a natural frequency of oscillation, you can interact with the system in the best way through resonance. And so you can send an electromagnetic wave with this frequency that is proportional to the field, and then you can get back a signal, and this signal basically allows you to make an image. So this is one of the techniques we use. So you can make very good anatomical images with this, but also you can probe how the brain works. Because basically, the hemoglobin, which is the molecule that transports the oxygen that you actually need for the brain to function, has different properties when it is linked to oxygen and when it is not linked to oxygen. So it is diamagnetic when it's linked to oxygen, and paramagnetic when it's not linked to oxygen. And so this means that when you have activity in the brain, if you have somewhere you have activity, you'll have an increase in the demand for ATP and then for glucose and oxygen. And then you will have the hemoglobin that has oxygen will liberate the oxygen, and you will have an increase in the deoxyhemoglobin in that region. And then when you have this decrease, this actionates a mechanism in the brain that will make that the blood and the volume, the blood flow and volume in that region increase. And they increase a lot, much more, and they bring a lot of hemoglobin with oxygen, the oxyhemoglobin, much more than what the brain can actually take. And so you have a peak in the signal here. And then when the activity returns to basal, the signal returns to basal. And so you can put a person in an MRI scanner, and you can subject this person to some experiment. And while you subject them to this experiment, you will measure a lot of images like the one I showed before. But you will look at each voxel of the image in time now. You can get, I don't know, 200 images while they're subjected to some experiment, to a stimuli, or they have to make a movement, or they have to think about something. And then you can measure that, and you can analyze that, and then you can make a statistical analysis to see which parts of the brain were more correlated to your experiment. And these using this signal that changes in the MRI. So basically, with this technique, you can have time series that tell you how the brain is working. The other technique I'm going to talk about is electroencephalography. It's the oldest technique that you can use to probe the brain functioning. And basically, you just put a lot of sensors on the scalp of the person that will measure the electrical potentials generated by the neurons. And you put these in a particular way. You have to measure the distance between this bone here and another bone here, and you separate these distances in 10 and 20%. And then you distribute the electrodes so that they will be more or less same over the same anatomical structures. And then you can measure basically voltages here related to some reference. And you can use these signals as well to probe the brain. So brain connectivity, you probably have been seeing this a lot. So I'm only going to talk how you can measure brain connectivity. So basically, we used 3 types of connectivity measures. One is anatomic or structural. Actually, these are 2 types of measures. The anatomic is where you actually look at the links, the physical links between 2 regions, which would be the white matter that I showed in one of the first slides, that how are 2 regions physically connected. This would be the anatomic. Or you can look at structural properties of the brain and try to find out how these properties are related to properties in another region. And then you have functional, which actually looks at the activity in a region and compares the activity between 2 regions. Or you can look at affective, which actually looks for a causal correlation between the activities of 2 regions. I'm going to talk only about these 2 types here. So for these, we use graphs that you've talked a lot about here, but basically, how you build these graphs, you can have, for example, the EEG time series or the fMRI time series, or you could have here, instead of a series, you could have a set of structural properties of a brain region. You always have to do a lot of pre-processing because these signals are very noisy. Then the EEG, once you do the pre-processing, you already have the time series. In the case of a functional MRI, you have to actually separate the brain into regions and extract the time series from the regions of interest. Once you have these regions, you have to set up a measure to compare these time series. And then when you compare this time series, you can build a connectivity matrix, where here you will have the regions, and each point here is the comparison between the series of 2 regions. And then you can or not binarize this matrix. And then you build, this is already your graph, okay? This is a weighted graph. This would be a binary graph. You can represent it like this or in any way like this. And then you can look at the properties, the topological properties of these graphs. And these you know a lot. So some types of graphs, undirected binary, directed binary, directed weighted. We actually use, in most of the works I'm going to show here, we use undirected weighted graphs. And these are some of the properties you can look at: degree, clustering coefficient, efficiency, and so on. So I will give you now examples of the type of things that we look at in the data. So I will look at one example using structural data. This is the work of student, PhD student of mine. So we got actually anatomical images. We separated these images in regions, and we looked at structural properties of each region that are called texture properties that are based in a matrix that is called the concurrence matrix that basically looks at statistical properties between pairs of pixels, of voxels actually. We extract texture parameters, and then we compare these texture parameters to build the graph. And so we built a graph, and we looked at the parameters that we obtained from this graph, and we found things like, we found network differences among men and women. We actually found a hub for women in the speech area, which was kind of funny. We found that some graph parameters showed a dependency with age, and we found that some particular structures in the brain, like the thalamus, that is responsible to get the senses that you measure and send them to internal structures, they had a differentiated texture. So another application that we've been working with are brain-computer interfaces and neurofeedback. So a brain-computer interface basically is a system where you measure directly the brain signals of a person, and you want to use these signals to control an external device. And you want to do that without using the normal pathways, which are basically what I'm doing now. I am thinking, and my brain signals are coming to my hand, and I'm moving this hand. I don't want to do this. I want to just measure this and then control, for example, the computer. So how do you do that? Obviously, you can't actually, not for now yet, you cannot actually read brain thoughts, the thoughts of the person. You have to measure the signal. You have to do a lot of signal pre-processing. And then you will do some sort of classification of the signal to say which command this signal is doing. And this has been used, and then you can give a feedback to the person or not to let them know if they are thinking the right way. Because obviously you have to devise strategies for the people to produce signals that are easily recognizable, which is not easy. And so this has been used in a lot of things. This idea started because some people wanted to measure people that had lost all the movement, so they couldn't communicate, because to communicate we use movement of the hands or of the mouth or whatever. And so they wanted to communicate with these people that were totally paralyzed. But this has been used, for example, this woman here, she has actually a sensor inside her brain, and she's using that to command this robotic arm. In this case, these sensors are near-infrared sensors. I didn't talk about this technique, but this is a case of a locked-in person, which is a person that lost all movement, and so the only way to communicate was through the sensors. And this is a case of EEG being used for rehabilitation, motor rehabilitation, which is one of the things that we want to use this for. So how do you make people produce signals that are recognizable? So the most popular strategies for that, one is called P300, which is a potential that appears 300 milliseconds after you see something that you are kind of expecting, but you don't know when it's going to happen. So how do you use that? It's a positive potential that appears in the EEG signal. So you can, for example, set up a keyboard like this, where all the letters, all the symbols are sort of dark, and they light up randomically. And so the person looks at the letter they want to write, and when this lights up, this potential will appear 300 milliseconds later, and because you actually know when the symbols light up, you can find out which letter lit up, and then you can write what the person is thinking. Another way is something that we call steady-state visually evoked potentials, which is that when you look at something that flickers with a given frequency, if you measure here in the occipital lobe the EEG signal again, you will find a peak in that frequency. So if you look at the signal, and they are looking here at something that flickers with 7 Hertz, you will find a peak in 7 Hertz and then in the harmonics. So you can have several options here, flickering with different frequencies, and then you will know to which option the person was looking at by looking at the Fourier transform and finding the frequency. And the other that interests us most in my work is called motor imagery, when you imagine the movement of a limb, you actually activate more or less the same regions that you activate when you actually move that limb. And this, in the EEG signal, you'll generate a signal that is called event-related desynchronization, which is that you have a decay, a decrease in the EEG signal in that frequency, in a given frequency. And if you look in fMRI, you will have actually activated more or less the same regions that you have with the movement. And so basically you can use this signal, look for this decrease, to find out if the person was thinking about moving one hand or the other hand. And for example, if I think about moving my left hand, I should have a decrease in the right hemisphere of my brain. And if I think about moving my right hand, I should have a decrease in the left hemisphere of my brain. So why is motor imagery interesting? Basically, it does not depend on an external device to deliver the stimuli, such as the other two options I showed. But for us, it's important because it activates more or less the same regions that the movement itself activates. It's being used for motor rehabilitation. So people that have a lesion, for example, a stroke patient that have a lesion in the brain, and they have to reorganize the brain to assume the lost functions. With this sort of training or of thinking about imagining, they can activate or they can reorganize their brain in order to improve the loss function. So, but what are the problems? We have a large intersubject variability, which means that the signals are very different when you go from one subject to another, but also you have a large intrasubject variability. So many times if I measure the subject this morning and I measure them in the afternoon, the signals will be different. For all EEG BCIs, the signal is very noisy. You have a lot of artifacts. So the main challenge in this area is to find discriminating and reproducible features, that is numbers, something that is constant and that you can use to actually make the system work. So because this is a field that is difficult, and we thought, okay, why the main features that are used are the power spectral density for this sort of thing. But we thought, what if we use connectivity measures? Because the brain never works by itself, only one region. It always works with several regions talking among themselves. So let's look at the connectivity. So we had an experiment where the person had to be at rest, imagine the movement of the right hand, rest, imagine the movement of the left hand, and so on. And we compared the EEG time series using a motive synchronization method, which is basically looking at the patterns in the EEG and translating the EEG signal into patterns like this, and then compare the patterns. And we found that, okay, if you looked at only one feature, the power spectral density was better, but if you looked at several features, we could actually use the graph metrics, and they were a bit faster to compute. So, okay, we managed to say that they are at least as good as. And then we wanted to look at how these features actually relate with the power spectral density, which is what we measure. And so we compare the graph features with the power spectral density, and we found that there was a larger correlation in the beta band, which is a bit of a higher frequency band. And we started looking at several similarity measures to compute this connectivity. So we looked at space-time recurrences, where we compare two signals, and you have a distance threshold to say if they are similar or not. And so you can build a connectivity matrix using this. We compare with other methods, such as Pearson correlation, Spearman correlation, phase coherence. This was a work done in collaboration with Diogo Soriano from the UFABC and his student Paulo Rodrigues. And then we found that the space-time recurrences were better than other frameworks, but all the frameworks were connectivity-based. And we have the eigenvector centrality that was the best feature regarding the processing time. And also we found the attributes in classical EEG motor cortex positions that are basically the positions right here in the motor cortex. We also look at another measure called core entropy, which is kind of a generalized correlation, but it uses kernels, and in this case we used a Gaussian kernel, and we compare it again to another methods, Pearson correlation and Spearman correlation. And these graphics show classification using two methods, which are the linear discriminant analysis and the extreme learning machine, which is a neural network. And the least error was with the core entropy, which is this smaller red here. And the combination of core entropy plus degree plus the extreme learning machine was the best thing. And we did that for two data sets, the one that we had collected and one that was from a public data set that is called BCI competition data set. And our results were comparable to the top 3 competitors. And this was done with a colleague from electrical engineering, Homies, and his student Luisa. This was done by another student of mine, Pedro. We wanted to compare again several methods of functional connectivity and see which ones gave the most discriminant and stable measures. And then again, we found that the beta band had more discriminant measures. In this case, we looked at some electrodes, and CZ, which is an electrode which is right here in the center of the head, was one with more discriminant measures. We compare 3 connectivity measures, which are connectivity methods, which were the motive synchronization, which was the best one, the weighted phase lag index, and the imaginary coherence. But one thing that we found that we were not very happy was that from the 10 subjects we looked at, only half of them, the most discriminating metric was also the most stable metric. So discrimination and stability were not kind of going together, not as much as we would have liked. So that's a problem. And this is another paradigm that another student of mine, Eduardo, has been working on, which is a different strategy to generate brain signals, which is about imagining speech. So in this case, people imagined 4 words, which were arriba, abajo, izquierda, derecha. They were in Spanish. It is a public database called Thinking Out Loud database. He used 2 different measures, which are strength, which is like weighted degree, and page rank, which is one measure that Google uses to rank the pages among themselves, the most important pages. The motive synchronization method, and in this case, we had 4 classes to distinguish, not only 2 like the other cases. I ended up not saying that, but in the other ones that we wanted to distinguish, imagining the movement of the right hand against imagining the movement of the left hand. And here we wanted to distinguish these 4 words. So we had better results, is this blue one here, than other 2 methods that use the same database. Okay, so this about BCIs, and what about neurofeedback? So neurofeedback training is a training that got popular in the last years. The idea is that if you train to produce a particular signal that is associated to some cognitive function, and you improve that signal, you will improve that function. It's kind of strange, but it works more or less in the same way than in a BCI. So you will make the person think about something, or give him or her some task to improve some cognitive function, for example. And then you will measure the signals, you will pre-process the signals, and then you will give a feedback to the person that can be the signal itself, or it can be usually some sort of a cartoon or something like that shows if the signal is going up or is going down, so that they know if they are having the right strategy. Because you say, you want the signal to go up or you want the signal to go down. So do something to make that happen. And then you will give this feedback. What is the difference between this and a BCI? In a BCI, the main goal is to actually control an external device. So for the BCI to work, basically, you either need the signal to be improved by the user so that you can easily recognize it with your system, with your computer system, or you need to improve your system, your classifier, so that you can recognize the user signal even if it's not that good. But in neurofeedback training, the main goal is the brain's autoregulation so that you can improve some skill or function, cognitive function. And so for the neurofeedback to work, the signal has to be improved by the user. You don't want the classifier to adapt. You don't want your system to adapt. You want the user to produce a better signal, because then, supposedly, they would have improved their function. So where is this being used? This is being used in pretty much everything, right? So if you can look here, you will find studies doing feedback treatment with attention deficit and hyperactivity disorder, depression, motor disabilities, anxiety, autism, obsessive-compulsive disorder, epilepsy, sleep disorders, and so on. Also for function improvement, motor function, executive function, memory, attention, focus, and so on. But the results are controversial, because most of these studies don't use what we call a control group, which is a group that does more or less the same thing without the feedback. And so it's hard to tell that the training actually caused this, or if you have a placebo effect or something like that. But specifically in these two cases, the results are less controversial. Actually, as I said before, when you think about moving a limb, you actually activate more or less the same areas, and this has been shown to improve this. And a lot of high-performance athletes think about their training to train as well. So we thought we would check this. And so this was a study of a student of mine who was already finished, co-supervised by my colleague by electrical engineering. And he did a really large and thorough work where he measured 30 subjects that had 10 sessions each. So he measured their brain signals 300 times, pretty much. He did a lot of things, but one of the things he did was he measured the EEG signals before and after, and during the sessions. And we also did fMRI scans before and after. They completed before the 10 sessions and after the 10 sessions. So he computed connectivity matrices, did all the pre-processing, and also he repeated a similar work, not exactly like this, because it was just one session and 5 cerebral palsy children. But when he did a stage in Spain for 1 year, and he worked there with Eduardo Rocon, who works with this type of children in Madrid. And what we found was that we had less consistency if you looked at, so here we had, in this case here, we had 3 groups. We had a control group that had no feedback. We had the active group that had the feedback correctly, which is we send the feedback based on their brain signals. And then we had a sham group, which was a false feedback. We gave a feedback to the person, but it was not based on their brain signals when they were actually imagining the movement. It was their resting state brain signals. And what we saw is that the active group, the blue is before the intervention, and the green is after the intervention. And this is looking at the similarity between the connectivity of all the subjects. And this similarity decreased after the intervention. So and this happened also for the children, the cerebral palsy children. And so what we interpret this is like each person devises a different way to cope with this task. And the more trained they are, more these tasks get specific in their brains. And so the similarity decreases after the training. The other thing we have been working on is extended reality systems and transcranial direct current stimulation for rehabilitation. So why use extended reality for rehabilitation? What's the idea behind that? So extended reality systems are either augmented reality or virtual reality systems. It's a terminology that encompasses all these things. And the idea is that you will have some system that will help you. These systems have been used a lot for gaming, okay? But why use this for rehabilitation? And the idea is that it has been shown that these sort of systems promote neuroplasticity and motor learning. So what is neuroplasticity? Is actually this reorganization that happens in the brain. And what's the idea? The idea is that these systems can trick the brain, and they give a false positive feedback. So what is the idea? If I have my arm paralyzed because I had a stroke, and I can't move it a lot, I can move it like this, but I see myself, my avatar in the game, and my avatar moves it like this. This gives a false positive feedback, which is a feedback that is false, but is actually positive because I see my arm moving more than I actually can. And this has been shown to actually promote a better reorganization of the brain. Apart from that, they are more fun because it's nicer to play a game than to do a repetitive movement all the time. And the most important thing, or one of the most important, is that they can be performed at home with remote supervision. So what's happening now? We have a global population that is aging. And so we have more and more incidence of diseases that will impair people, such as Parkinson and a lot of diseases like that, stroke. And so more and more people actually need rehabilitation, motor rehabilitation. And we don't have enough professionals or clinics to attend this demand, especially with the years that come. So it is important to, so with these sort of systems, you could have one professional that would be at home supervising several patients that would bring their system. Obviously, it couldn't be a system like this, it's very expensive, but something cheaper. They could bring the system at home, and they would do the rehabilitation remotely supervised by a physiotherapist, for example. So what are the XR tools that we investigated? So this one was developed by Alexandre Brandão and Diego Dias. Alexandre Brandão developed the first version of this system at UFSCar, the Federal University of São Carlos. And then he came to do a postdoc with me. And so this is basically a system where you walk along the street view places of Google Maps. So it's kind of good because you can choose the place where you can walk. You have an avatar that walks there. So you do a stationary march. So it's a lower limb rehabilitation tool. And this is a puzzle, basically, where you can put any picture you want here, and you will separate the picture in places, and then you have to bring the places with your hand. And so it's upper limb rehabilitation. Then, and this is a tool to measure the positions of the joints so that you can give a feedback to the physiotherapist of how the movement is improving over time. You can make plots and stuff of how the range of motion of the patient is improving. This is another system that we evaluated that was developed by Gil D'Assis at University of São Paulo, and then she also came to do a postdoc with me. And this actually did exactly what I was telling you about the false positive feedback. They filmed the patient, and then they replaced the paralyzed arm of the patient with a virtual arm. And so this arm that was virtual would move much more than what the actual arm of the patient could move. Another tool that we use was developed by the group of Paul Verger. He was at Barcelona at the time. Now I think he's somewhere in Belgium or Holland. I don't remember. This is called the rehabilitation gaming system. It's also an upper limb system for rehabilitation, where you have an avatar of the hands here that have to catch the balls. They have several games that are related to reaching and moving these balls. An important thing about this system is that in real time, it adapts to the patient. So if the patient has a lot of difficulty, the game is, and the game realizes that the patient is not getting right the goals, it diminishes the speed of the balls and diminishes the difficulty of the game. If they are getting everything right, then it increases the speed. So it adapts to the person, so the person doesn't get bored or doesn't get frustrated because they cannot do anything. And the other one was developed by Elena Partesotti, who is an art student. She was actually supervised by Jonatas Manzoli from Unicamp at the Arts Institute. and I co-supervise her. Now Jonatas is retiring, so I became the main supervisor. And they developed this game. This is a cave where you have several projectors and several sound systems. And then when you move here, you produce sounds and colors. And these sounds and these colors depend on the movement that you do, that you make. So we evaluated the system NeuroR from Gilda. In this case, this was the first study we did. We were only able to measure 3 patients. We did fMRI before and after. They did 8 sessions with the tool. And what we found for 2 patients was a significant increase in functional connectivity in motor regions. For 1 patient, which is actually this one, these changes were not significant. And we also had measures of the range of movement, and they seemed to improve for the 2 patients that had significant changes. We also did for the gesture collection, that system. This was only the upper limbs part, which is that system that my first postdoc developed. We had at first 14 patients, 7 from experimental group that actually did rehabilitation. They did standard rehabilitation plus the rehabilitation with gesture collection, and the other group did only the standard rehabilitation, but for the same amount of time. These numbers in brackets is because we lost some data because the patients the data wasn't good. That's the problem with the MRI data. They had a lot of movement, and so we had to discard some data, so we ended up only with 5 people in each group. And so we measured functional connectivity. This shows the connectivity changes, and these are the significant changes. And so we had more increases in functional connectivity in the experimental group compared to the control group. And one important thing, both groups, they improved in the clinical scales, but we had the more increases in functional connectivity in the experimental group. And the changes were different. So the experimental group, they had changes in regions more connected to reward-based motor learning. And the control group had changes in regions more related to purely mechanical activity. So this is the changes due to RGS. So we only measure 8 people, and we have to discard 3 because of bad data. All these works actually were interrupted by the pandemic. So the idea in this work specifically was to measure 30 subjects. I'm not saying here, but each subject has to come and do several rehabilitation sessions. So in this case, there were 12 rehabilitation sessions done twice a week. So it was like a month and a half, more or less, for them to complete the whole thing, and then measuring them before and after. So in this case, we just had that all subjects show improvement in the clinical scales, and we had the significant variations were mainly increases, again, of functional connectivity. And this is the work with the Be Creative, which is that tool that was kind of different. This was the only work done with actually healthy subjects. So we started with 11, but we couldn't complete. It was also interrupted by the pandemics, the COVID pandemics. And so we ended up with complete data only for 5 of them, because the other 11 didn't finish the protocol. And we had some different results in the sense that instead of showing increases, the subject had decreases in functional connectivity, which was different from what we obtained with the stroke patients. And also we had an emotional response, which was different from the other ones, but possibly because of the type of relation with the tool that was different. The other ones had very specific tasks to complete, and this was very artistic. So finally, the last study we have been doing is using transcranial direct current stimulation. So this is basically, the idea is that you will deliver some very low current on the scalp of the person, 1 to 2 milliamperes, for about 20 to 30 minutes. It has a limited side effect, so it's relatively safe. And if you place the anode in the lesioned cortex, or in the place you want to put, and then you place the cathode in a neutral place. Then the anodal TDCS that we call, it has an exciting effect. It increases the neural excitability, and the cathodal increases the neuronal inhibition. And so for motor rehabilitation, you could either use excitation of the cortex that is lesioned, or you can use the inhibition of the cortex that is non-lesioned. And we actually use this one here, the first one. So this is a quite complex project because we work with chronic stroke patients. So these are patients that had the stroke at least six months before, previously. So they come, and we divide them in either one of these two groups. One will do XR rehabilitation plus the ATDCS, and the other one will do the rehabilitation, the XR, plus a sham signal. So they think they are actually having their cortex excited with current, but they are not. But we put all the equipment and everything, and we have a start current that actually goes to 0 after 30 seconds. And they come, and for 10 sessions, so they come on a Monday, and they come Monday to Friday and Monday to Friday for 2 weeks. And we measure them on Monday, on the following Friday, and on the last Friday. And all the measurements we do are clinical and functional scales. We measure the range of motion using that software that I told you about before. We measure EEG, we measure EMG, which is electromyography in the muscle. And I have this very large team, which is composed of some physiotherapists, doctoral students, undergraduate students, doctoral students, postdocs, and a lot of people. So I show here only the results that we have. We are about to publish these results. We took 2 years to collect data from 19 subjects because they come for 2 weeks. We can only measure one patient at a time because of restrictions of the lab and of personnel. And also there were some weeks that we couldn't find patients to come and do the rehabilitation. So they had 10 sessions of 30 minutes. They did the EEG, where they had to imagine, they had some resting state, and they had to imagine the movement of one hand or the other hand. This was the experimental setup. This is actually a result that has not got to do with functional connectivity. This is just a symmetry index, where the larger the symmetry index, the more asymmetric the brain is. So obviously, if you have a lesion in one side, you would expect the brain to be really asymmetric. When the lesion starts to heal, or you have the area around the lesion that gains the functions of the lesion area, then the brain becomes more symmetric. So we can see here that the TDCS group had a decrease in asymmetry, so they increased symmetry in some of the frequency bands, and the sham group sort of stayed more or less the same. In some patients, increased their asymmetry. You calculate looking at the power spectral density of the region of some specific electrodes. We use basically the electrodes over the motor cortex in the hands area. And these are some measures from strength, which is the weighted degree. So this is the global strength. So we can see that there wasn't much change for the TDCS group here in the rest. But there was some decrease in the sham. We actually don't know yet how to interpret these results. And here, we again didn't have much change for during the imagination of the paretic hand, and we had a decrease for the sham group. And looking specifically at the strength over the hand region, ipsilateral means looking specifically over the lesion. we had some increase in functional connectivity for the TDCS group and decrease for the sham group. And here again, but these bands also, they are not usually associated, these frequency bands, I didn't talk much about them, but they are not usually associated with this type of thing, so it's strange. And here we sort of had some decrease, and here some increase, and here some decrease. And so, what are the difficulties and limitations of this sort of work? As I mentioned, the samples used in each study, particularly the XR ones, were small. We wanted to recruit more subjects, but we did have the problem of the pandemics that interrupted at least 3 of the studies I showed. But also because the patients have to come and do a large number of sessions. So they stay 2 weeks with us. They have limited mobility, which means that they depend on someone to bring them. They have to skip work to bring their relatives to do the thing. So it's kind of difficult. And we have the problem with the data quality, the MRI exams. Some of the exams of the patients were not good because they moved or something during the exam. We have heterogeneous samples, so we are looking for patterns where it's really hard to have patterns because the sample is not really homogeneous. It's hard to find large number of patients with the same lesion location and the same clinical condition. The lesion will not always be exactly the same, and the impairment will not always be exactly the same. And so obviously, it's difficult to find patterns. We have dependence on health professionals. We cannot do this work without physiotherapists, that they have to accompany the patients, and they have to, all the clinical evaluations must be done by health professionals. And it's hard to be in a physics institute and get financing for a physiotherapist. So, well, we manage, but it's not simple. But the conclusions are, so regarding BCIs and neurofeedback, the connectivity measures were related to motor imagery event-related desynchronizations. The connectivity measures produce results at least as good as the power spectral density for motor imagery paradigm, and better results for the inner speech paradigm. And the feedback resulted in more different connectivity patterns among subjects, both for healthy subjects and cerebral palsy children. Regarding the rehabilitation with XR or XR plus TDCS, most patients showed improvement in clinical scales, so this was at least something good because most of the patients improved in something. The XR plus conventional therapy, in the case of the gesture collection, enhanced the treatment effect. And in this case, the XR plus TDCS, we found a decreased asymmetry for TDCS, for the TDCS group in higher frequency bands. And we found an increased connectivity for TDCS and a decreased connectivity for the sham group in bands not usually associated to movement, execution, or imagination. But in general, we found brain changes pointing towards restoration to normal patterns. And I want to thank all past and present students because this work wouldn't be possible without them. All collaborators, I have a lot of collaborators from all these projects I showed at the beginning. All subjects, healthy and patients, and all the Brazilian Federal and São Paulo State research funding agencies. And thank you.
Thanks, Professor. Now we have time for some questions.
Thank you, Professor, for this great presentation. I would like to know about the data from your lab. Is it available, and where can I find it?
It can be made available for now. We're still working with a lot of data that we have collected, but yes, we can talk. We can talk about the collaboration, and we can share the data. Yeah, sure. Send me an email. My email is, I didn't put it here. No, my email is easy. Gabriela, with one L, @unicamp.br. So just send me an email.
Yeah, hi. So my first question was actually the same, if you have data available. And then my question is, like, when you talk about functional connectivity, it's like Pearson product correlation, which you have measured, that you are calling functional?
No, functional connectivity is Pearson correlation when you talk about MRI, fMRI data, because other similarity measures have been used, and they found that they were at best the same as Pearson correlation.
Okay.
So because Pearson correlation was simpler,
Yes,
is
yes,
established.
yeah,
So in fMRI, that's established. But in EEG, it's not established. So
yeah,
the functional connectivity, we have been using a lot this motive synchronization method. But we have used other things. So the last studies here with the TDCS work we have done with imaginary lagged coherence. We have used a lot of different measures. So weighted phase lag index.
Okay, I see. Have you ever tried like this, all this mutual information kind of measure for functional connectivity?
We haven't tried mutual information yet. Mutual information has been tried in fMRI, and it was discarded because it didn't. It was a lot of work for this more or less the same result. But no, we haven't used mutual information.
How many nodes you had for the EEG data? So how many?
So we have. Yeah, I didn't talk about that. Different studies had different amount of nodes. In these last studies with TDCS, unfortunately, we had a very, very nice EEG equipment with 64 channels that broke in the middle of the pandemic. So now we have a 16-channel EEG, which is the one we have used in the last experiments. We are hoping to get a new one with 32 channels soon in that DREAMS project. But it depends. Like the inner speech work was, I think, is not our data, is public data. Is 64 or 128 channels, I think. So that can vary a lot.
I'll get back to you on data later, perhaps.
Yes, my data is, all the data from the neurofeedback studies is, not all, 2/3 is 64 channels and 1/3 16 channels, and most of the last data is 16 channels.
Thank you.
Questions?
I have a question. I'm curious, why did you choose that amplitude for the TDCS? You have why did you choose that specific amplitude? Did you not change the amplitude in the patients? You choose this fixed amplitude?
The amplitude of the current.
Yeah, exactly.
It has been. I wouldn't try, because we are not trying to. We are using methods that are established. The thing we are doing is looking at the brain changes. I wouldn't try to increase the current because I'm not doing an experiment on TDCS, specifically on the parameters of TDCS. I wouldn't dream of increasing the current because I don't know what effects it will have in the brain of the people. So the maximum current that has been used, there are works obviously that have tried more things, but the usual is between 1 and 2 milliampere. And I think we are using 2 milliampere or 1. I don't remember by heart now. I think it's 1 or 2 for 30 minutes. And
okay, just to complement, and about the alternating
the
current.
The microphone like this.
Okay, thank you. And about the alternating current, do you think that you can try?
There are works with alternate current, but we haven't used that yet. So there are some works that are called, is that repetitive to this? No, this is another one. There are works with alternate current. There are, but we haven't tried that yet.
Okay, thank you.
I also have a question about how you collect the data. For instance, I was thinking about when you analyze things like the motives. The point is, are the motives independent on how you sample the data? That is, the frequency of the sampling of the data. Is the motive independent of that? Because
No.
It's not. Is it
No,
independent or it's not?
It's not. It's not independent.
It's not independent.
No. Well, we actually look. I didn't enter into details here, but most of the studies we look at the signals in different frequency bands. So either we look at the usual EEG frequency bands, which are alpha, which is from 8 to 12. This is one of the main bands we look at because alpha and beta bands are the main bands that are related to motor execution and motor imagery. So alpha is from 8 to 12 hertz, beta is from 13 to 31 hertz, more or less. So these are the main frequency bands that we look at. So sometimes we just look at this frequency band from 8 to 12 and from 13 to 31. Sometimes we look at lower frequency bands, like in this last case, which is the theta, delta, which are like from 1 to 4 hertz, from 4 to 8, 8 to 12, and so on, and the gamma band, which is above 31. Sometimes we look 2 by 2 hertz, so 2 to 4, 4 to 6, 6 to... And you obtain different motif sequences for each band, obviously. Yes, because you're suavizando o sinal, you're smoothing the signal, right?
Yeah,
so
yeah.
the motifs will be different. Yes.
Yeah, I can see, but shouldn't be some events. Let's say, I'm just thinking. For instance, if you are talking about an epileptic crisis, I mean, the event of the crisis should be more or less independent of the sampling, shouldn't it? I mean, I'm not talking about 2 hertz, which is too low, but I'm talking about, I mean, these people from could have the EEGs of epilepsy usually are about 200 hertz or something like that. So those are high frequency samplings, but let's say I think that a crisis should be. To see a crisis should be more or less independent. On the other hand, the motives, if you take too high a frequency, you are going to get a lot of garbage in the motives. That's what I'm saying.
In regard of epilepsy, to see a crisis, it depends on which type of event you're looking in the signals.
Yeah.
If you have the spikes,
Yeah.
well, you have infinite frequencies there, depending on how you sample, and it will change depending on how you sample.
Yeah.
It will change the motif. But you have some larger patterns in epilepsy that are not the spikes specifically. So they will change again if you change the frequency. So yeah, you will be able... I don't know, because the neurophysiologists, they actually look at the signal in the sampling frequency that it was acquired, but they don't look at different frequency bands. That's something that we do because in our work specifically, in this work specifically, that we are looking at motor-related patterns, we are more interested in the alpha and beta bands. Although in this last work, we found things outside of these bands. So sometimes we look at other bands, but usually we are interested in these ones. But a neurophysiologist, when they look at the signal, they look at the signal without filtering, in bands at least. So, and they don't do a mathematical processing. So.
Thank you, Professor, for your presentation. My first question concerns your techniques of data collections. So we know that a magnetic field has the property to modify the behavior of neuronal cells.
Can you
magnetic
repeat?
field
Yes.
has the property of modifying the behavior of neuronal cells. So you use modify the behavior
of neural cells. Okay.
Yes. So you use the magnetic resonance to establish the functional connectivity. So do you think this magnetic field couldn't insert some difference?
Not really, because we are measuring very specific things. What we measure in magnetic resonance imaging is actually the changes in local fields that you will have because of the properties of oxy and deoxyhemoglobin. So, okay, when you put the person inside the scanner, the magnetic field is static. Okay, but you have to switch on and off all the gradients and stuff. But
yes,
you're actually looking at how the signal will decay differently, will relax in a different way, because when you have these paramagnetic or diamagnetic molecules, they will interact with the field surrounding, only the paramagnetic ones actually,
yes, yes,
and will change the local field, and that will make the signal relax faster.
yes.
So that is basically what you're measuring. So I don't know if I got your question.
We will discuss after. My second concern the period of rehabilitations. We know that during the learning process, some connections are established.
Mm-hmm.
And I want I want to ask you if during the rehabilitation, because we are here beside a patient,
Mm-hmm.
during the rehabilitation, is connection also established?
Yes, that's the idea. The rehabilitation is a type of learning. It's a type of learning.
Even some cells are damaged?
No, yeah, but you will create new connections in the healthy parts of the brain. So basically the cells, usually the neurons that surround your lesion, that the cells that are damaged, these, forget about them, they are dead. But you expect that the neurons surrounding that region will take up the function of the cells that were lost. And these ones, yes, they will establish new connections.
Oh, so you
So that's what we call neuroplasticity or brain reorganization.
Yeah, yes. So you are saying that without caring about the age of the patients, neurons are able, the age,
Independent of the age.
It depends of age.
Mm-hmm.
Of if you have the old man, their neurons be able to establish connection even at the old age?
Yes, they are.
Okay.
Obviously, it's easier the youngest the person is, the easiest it is. So for younger people, it's easier to make these new connections. For older, the oldest the person gets, the more difficult it is to make these new connections. But this process of reorganization, it happens throughout all life.
Okay, thank you very much. I'm also interested by your data.
Yeah, okay.
I think you can take. Thanks, Professor Gabriela again.